Welcome back to the Deep Dive. Today we are taking a stack of biochemistry sources and mapping out one of the most fundamental processes that underpins all cellular life, how we handle glucose.
Our mission is to systematically navigate and really get to grips with the dual metabolic pathways of glucose. So on one side, you have glycolysis, which is the burn phase, and on the other, gluconeogenesis, the build phase.
Right. And we're going to treat this as a complete guide to how your body manages its most essential fuel, pulling directly from the foundational biochemistry material we have in front of it.
Exactly. And the central theme here, the thing that's so elegant, is this incredibly fine-tuned reciprocal control.
Let's start with glycolysis. This is the ancient pathway. It's a sequence of, what, 10 reactions?
10 reactions that take one 6-carbon glucose molecule and splits it into 2 molecules of pyruvate. And in the process, you get a net profit of 2 molecules of ATP.
And the key, as you said, is that it's ancient. It's fundamentally anaerobic.
Absolutely. It evolved long before oxygen was a major player on Earth, so it doesn't require O2 at all. This makes it the perfect pathway for generating that instant burst of energy when, the demand for power outpaces your oxygen supply.
And the counterbalance to that is gluconeogenesis.
Gluconeogenesis, which is the synthesis of glucose, but starting from non-carbohydrate precursors.
So we're talking about molecules like lactate or certain amino acids from protein breakdown.
Or even glycerol from your fat stores. The body can take those and build a brand new glucose molecule from scratch.
The relationship between these two is what's so critical. They look like they're just reversals of each other, but they're not.
Not at all. I mean, they do share many of the same enzymes, but the 3 key irreversible steps in glycolysis have to be completely bypassed by new, unique reactions in gluconeogenesis.
And if that control system failed, if they were both running at the same time,
it would be a disaster. You just hydrolyze ATP, generating heat and wasting a massive amount of energy. It's called a futile cycle for a reason.
So why is glucose so vital? Why do we have this huge, complex, frankly, energy-consuming system just to keep its level stable? What's the physiological imperative?
The so what here is immense. I mean, glucose is the sole fuel source for your red blood cells.
Because they don't have mitochondria.
They have no mitochondria. And critically, under normal non-starvation conditions, it is the primary fuel for your brain. If your blood glucose drops significantly, the entire organism starts to fail.
Let's make this more dynamic. Think about a sprinter, like Usain Bolt, exploding out of the blocks for the 100-meter dash.
Perfect example. His muscles are firing so rapidly, the demand for ATP just completely outstrips the rate at which oxygen can be delivered to the muscle tissue.
So he's in an anaerobic state.
Totally anaerobic. In that moment, the muscle relies entirely on glycolysis. Glucose is metabolized like lightning, and since there's no O2 to process the pyruvate further, the pyruvate gets converted to lactate. It's a quick, dirty 2 ATP yield, but it's enough to keep that sprint going.
And you compare that to, say, a long, slow-distance run.
It's a different world. Oxygen delivery is perfectly adequate. In that case, the glucose is metabolized far more efficiently. The pyruvate goes to the mitochondria and gets completely oxidized to CO2 and water, which yields way more energy per glucose molecule. The 2 pathways allow you to survive in both conditions.
And this entire field of knowledge, this incredibly detailed mechanism, it all stems from a completely accidental discovery.
It does, back in 1897 by Hans and Edward Buchner.
What were they even trying to do?
They were trying to purify yeast extracts for, of all things, possible medicinal purposes. They needed a preservative that wouldn't kill the very thing they were trying to study, so they chose sucrose sugar.
And they mixed this yeast juice, so literally the contents of the yeast cells without the walls with the sugar, and it just started bubbling.
Exactly. They were completely stunned. This cell-free extract was rapidly fermenting sucrose into alcohol. And you have to remember, up until that point, the dogma from Louis Pasteur was that fermentation was tied to the vital force of a living cell.
So this accident completely shattered that idea. It proved the chemical machinery, the enzymes, could work perfectly fine in isolation.
It basically launched modern biochemistry. It showed that life's processes are governed by chemical principles, and then later work on alcoholic fermentation in yeast and lactic acid fermentation in muscle showed this underlying unity.
That many of the steps were the same.
Most of the intermediate reactions were identical. It confirmed that this fundamental 10-step pathway is an ancient, unified piece of cellular hardware.
Okay, let's unpack the core pathway itself then. Before glucose even gets into those 10 steps, we have to talk about digestion and, I think, a more basic question. Why glucose? Why is it such a favored molecule?
Well, our diet usually provides these big complex carbs like starch and glycogen. Digestion kicks off with amylase, an enzyme that cleaves the main 1,4 bonds.
But it leaves behind these branch molecules, right? Limit dextrins.
Right, because the As1-Sec6 branch points resist the amylase. So then at the surface of your intestinal cells, you have this whole team of specialized enzymes, maltase, sucrase, lactase. Their job is to finish the process, breaking everything down into absorbable monosaccharides,
Glucose, fructose, and galactose.
which are then transported into your bloodstream. But to your question, why does the body favor glucose over the others? The answer is all about its structure
And safety, it seems.
and safety. All monosaccharides can exist in two forms, a stable ring and a reactive open chain form. In that open chain, the carbonyl group can non-enzymatically attack amino groups on proteins. It's a process called glycosylation.
And that's bad. That damages the proteins, potentially creating these things called advanced glycation end products, or AGEs.
Exactly. Glucose minimizes this risk. It has a much stronger tendency than other sugars to stay in its most stable ring conformation, specifically the bucose form where all the hydroxyl groups are in the low-energy equatorial position.
So it's just less likely to be in that dangerous reactive open chain form.
It's an evolutionary safety choice. It's the safest sugar to have circulating at high concentrations in your blood.
That is a fantastic example of structure dictating function. Okay, let's jump into the 10 steps of glycolysis, all happening in the cytoplasm. We divide them into two stages. Stage 1.
Is the investment stage. It's going to cost the cell 2 ATPs. The goal here is to trap the glucose, destabilize it, and then cleave it efficiently.
So step 1, the trapping step. This is catalyzed by hexokinase. It uses 1 ATP to phosphorylate glucose.
Turning it into glucose 6-phosphate, or G6P, and the consequence of this is immediate and critical.
The phosphate group.
That new phosphate group has negative charges,
Hmm.
and those charges prevent the G6P from being transported back across the hydrophobic cell membrane. It effectively traps the glucose inside the cell. It's committed now.
Hexokinase itself is a classic example of this induced-fit mechanism. Can you break that down? Why is it so important?
So hexokinase, like all kinases, uses ATP, but ATP is also really susceptible to just being broken down by water, which would turn the enzyme into a wasteful ATPase.
All right, you don't want that.
To prevent this, hexokinase has two lobes with a cleft between them. When glucose binds, the whole enzyme changes shape. The two lobes swing inward and close that cleft.
So it's a physical barrier.
It's a physical barrier that does two things. First, it aligns the glucose and ATP perfectly for the reaction. And second, and this is maybe more important,
It kicks out the water.
it excludes water molecules from the active site. By creating this non-polar environment, it physically stops water from attacking the phosphate, ensuring that precious ATP is only used to phosphorylate glucose.
Brilliant. Okay, step 2 is a simple isomerization. Phosphoglucose isomerase turns G6P into fructose 6-phosphate, or F6P, and then comes step 3, the second investment and the most important regulatory checkpoint.
Phosphofructokinase, PFK.
PFK uses a second ATP to convert F6P into fructose 1,4,6-bisphosphate, or F16BP. This is what we call the committed step.
Right. Once that molecule is phosphorylated at both ends, it is absolutely destined for breakdown. There's no turning back.
And that isomerization in step 2, changing it from a glucose to a fructose structure, suddenly makes a lot of sense when you look at F16BP. Why do that?
It's all about efficiency and symmetry. If you just added the second phosphate to the original glucose structure, the cleavage later on would give you two unequal fragments, a 2-carbon and a 4-carbon piece.
Which would be a metabolic nightmare to deal with.
You'd need two completely separate complex pathways. By changing to the fructose structure first, the molecule is perfectly symmetrical, so it's prepared for step 4.
Step 4 is the cleavage, catalyzed by aldolase. It splits F1416BP into two 3-carbon units.
Glyceraldehyde 3-phosphate, GAP, and dihydroxyacetone phosphate, DHAP.
Now we have a problem, right? Only one of those, GAP, can continue down the pathway.
That's right, so half the molecule, DHAP, would be wasted. Step 5 is the salvage operation. It's catalyzed by triose phosphate isomerase, or TPI, which rapidly converts DHAP into GAP.
And this enzyme, TPI, is famous for being kinetically perfect. It works almost instantly. But there's a fascinating detail about how it maintains safety while being so fast.
It is fascinating. TPI works through a really unstable intermediate called an anediol. And in the watery cytoplasm, this intermediate is really prone to decomposing into useless, even toxic side products.
So the cell has to prevent that from happening. How does the enzyme contain it?
It uses a structural defense. When the substrate binds, a flexible loop on the enzyme acts like a little hinged lid. It just closes over the active site, completely trapping that unstable anedial intermediate.
So it holds it securely until the reaction's done.
It holds it just long enough for the isomerization into GAP to complete, physically preventing any side reactions with water. Then the lid opens and it releases the final product. It's this beautiful example of an enzyme evolving for defensive accuracy, not just speed.
And that wraps up stage 1. We spent 2 ATPs, but now we have 2 molecules of GAP ready for stage 2.
The energy generation phase. Now we're in the payoff stage. We're going to net 4 ATPs and 2 NADHs from these 2 GAP molecules.
Step 6 is the conversion of GAP to 1.3 bis-phosphoglycerate, or 1,3,3-BPG. The enzyme is glyceraldehyde 3-phosphate dehydrogenase, GAPDH. This is a huge step. You have oxidation, NAD plus reduction, and you're adding an inorganic phosphate. How does the cell couple a good reaction with a bad one here?
This is just a beautiful piece of chemical engineering. Inside GAPDH's active site, there's a reactive cysteine residue. So first, the aldehyde group of GAP is oxidized to a carboxylic acid. But, and this is the key, instead of floating away, that acid gets temporarily linked to the enzyme.
Through a high-energy bond.
Through a very high-energy covalent bond called a thioester intermediate. So the energy that was released by that favorable oxidation is now chemically stored in that thioester bond.
I see. And that stored energy is then used to drive the next part.
Exactly. It's used to drive the unfavorable formation of the high-energy acyl phosphate product, 1,3-BPG, when inorganic phosphate comes in and attacks that thioester. It's essential. Without that intermediate, this wouldn't be energetically possible.
And 1,4,3-BPG is the first big payoff because it has this high phosphoryl-transfer potential. In step 7, phosphoglycerate kinase transfers that phosphate directly to ADP.
And you get your first molecule of ATP plus 3 phosphoglycerate.
And since we started stage 2 with 2 GAP molecules, we're actually getting 2 ATPs here.
Right. And this is what we define as substrate-level phosphorylation. You're making ATP directly from a high-energy substrate, no proton gradient involved, and these 2 ATPs have now fully paid back the 2 we invested in stage 1. We're at break-even.
The next 2 steps are all about rearranging the molecule to create another, even higher energy product. Step 8 uses a mutase to shift the phosphate, forming 2-phosphoglycerate.
In step 9, the enzyme enolase performs a dehydration. It removes a water molecule to create phosphoenolpyruvate, or PAPE.
And PP has the highest phosphoryl-transfer potential we see in this whole pathway. Why is this molecule such an enormous spring of energy? It looks like a simple phosphate bond.
The reason is trapped instability. PEP is basically an unstable molecule where the phosphate group is trapping it in this very reactive enol form. The second you remove that phosphate in the next step, the molecule spontaneously and almost instantly converts to its superstable keto form, which is pyruvate.
So it's the massive energy difference between the unstable enol and the stable keto that provides the driving force.
A huge driving force. The Gibbs free energy change is something like minus 62 kilojoules per mole. That's what's needed to synthesize that final ATP.
Which brings us to step 10, the final profit-generating step, catalyzed by pyruvate kinase. PAP plus ADP gives you pyruvate plus ATP.
And that gives us 2 more ATP molecules. That is our net profit for the entire pathway. So the final tally from one glucose is 2 ATPs, 2 NADHs, and 2 pyruvates.
But now the cell has an immediate problem, those 2 NADH molecules. We have to talk about regenerating NAD plus E. The cell only has a finite supply of NAD plus lin, which comes from vitamin B3, niacin.
Right. If you don't regenerate NAD plus an inlet, step 6 of glycolysis, the GAPDH step, grinds to a halt. The whole pathway stalls.
So the fate of pyruvate is all about solving this redox problem. This is fermentation.
Fermentation, exactly. It's an ATP-generating process where an organic molecule acts as both the electron donor and the electron acceptor.
So, fate 1, alcoholic fermentation.
Yeah.
This is what yeast does. Pyruvate is first converted to acetaldehyde, which releases CO2, and then the acetaldehyde is reduced by NADH to form ethanol.
And that reduction step is what regenerates the NAD plus AC. The final enzyme here, alcohol dehydrogenase, has a zinc ion in its active site, which polarizes the carbonyl group of the acetaldehyde, making it much more reactive. The net result is you've consumed glucose to get ethanol, CO2, and your 2 ATP.
Fate 2 is lactic acid fermentation. This is what happens in our own bodies during intense exercise or all the time in our red blood cells.
Here, lactate dehydrogenase catalyzes the direct reduction of pyruvate by NADH to form lactate. And once again, you've recycled your NAD plus X.
And this is how your muscle gets those short bursts of ATP, The buildup of lactate is what lowers the pH in the muscle, which contributes to fatigue.
And that low pH is also a signal. It actually feeds back and tells the main regulatory enzyme, PFK, to slow down.
It's incredible. We just discussed three totally different dehydrogenases, JPDH, alcohol DH, and lactate DH. They do different things to different molecules, but they share a nearly identical structural feature.
That's one of the most compelling pieces of evidence for common evolutionary ancestry in biochemistry. They all share what's called the Rossmann fold.
Which is a structure for binding NAD+.
Ricimet, it's a highly conserved structural motif specifically for binding that dinucleotide coenzyme. The fact that it's in all these functionally distinct enzymes strongly suggests that a primordial gene for a dinucleotide binding domain was just copied and reused over millions of years. It really highlights the modularity of metabolism.
Speaking of modularity, the body doesn't just run on glucose. What about other common sugars like fructose and galactose? How do they plug into this system?
They all have to be converted into one of the glycolytic intermediates. Fructose metabolism mostly happens in the liver. It's a separate pathway that uses fructokinase to make fructose 1-phosphate, which is then cleaved into DHAP and glyceraldehyde.
And that glyceraldehyde gets phosphorylated to GAP.
So both products are now deep in the glycolytic pathway. What's the problem with too much fructose then? Why is it linked to things like fatty liver disease?
The problem is that the fructose pathway completely bypasses the main regulatory checkpoint, the PFK reaction.
It sneaks in after the gatekeeper.
It totally bypasses the gatekeeper. Since PFK is what filters the flow of carbon based on the cell's energy needs, bypassing it means you have this unregulated high-speed flow of intermediates straight to pyruvate and then into the mitochondria to make acetyl-CoA.
And when you have way too much acetyl-CoA for your energy needs, the liver has only one option for it.
It converts it into fatty acids. And this unregulated flood of carbon directly into fat synthesis is why high fructose intake is so strongly linked to fatty liver and insulin insensitivity. The system just wasn't designed for that. What about galactose, the sugar in milk?
Galactose gets converted to G6P in 4 steps. It's a clever little cycling mechanism that uses UDP glucose as a carrier to ultimately flip one of the hydroxyl groups on the galactose molecule.
And the pathology associated with this, classic galactosemia, is caused by a deficiency in one of those enzymes.
Right, the galactose 1-phosphate uridyl-transferase. If that enzyme is deficient, galactose builds up. The body then reduces it using aldose reductase to a sugar alcohol called galactitol.
And galactitol is the real problem.
It's the problem. It can't be metabolized, and it can't easily exit the cell, so it just accumulates in tissues like the eye lens. To deal with this high solute concentration, water rushes into the lens to maintain osmotic balance, which causes the swelling and clouding that we call cataracts.
So we know the steps. Now let's talk about the choke point. Glycolysis isn't just about ATP, it also provides precursors for biosynthesis. To manage these conflicting needs, the pathway has to be tightly controlled at those 3 irreversible steps.
Right. Hexokinase, PFK, and pyruvate kinase. And the control mechanisms are completely different depending on the tissue's job.
Let's start with skeletal muscle. Its only purpose for glycolysis is fuel for contraction.
So regulation here is all about the energy charge, the ratio of ATP to its siblings, ADP and AMP.
PFK is the main pacemaker. High levels of ATP will inhibit PSK.
Right. The ATP binds to an allosteric site, not the active site, and it decreases the enzyme's affinity for its substrate, F6P. So when energy is abundant, the pathway slows down.
And conversely, AMP is the strongest activator. What makes AMP such a potent signal?
It's all about concentration and magnification. There's an enzyme called adenylate kinase that maintains an equilibrium. 2 ADP molecules can become 1 ATP and 1 AMP.
I see. So because ATP levels are normally really high and AMP levels are normally tiny, even a small percentage drop in ATP gets magnified into a huge percentage spike in AMP.
Exactly. A 10% drop in ATP might lead to a several 100% increase in AMP. So AMP acts as this incredibly sensitive emergency signal. It allows the muscle to respond instantly and crank up PFK activity when energy starts to run low.
And we also saw that low pH from lactate buildup inhibits muscle PFK.
It's a protective mechanism. It stops the muscle from accumulating too much acid.
Upstream from PFK, hexokinase is inhibited by its own product, G6P. This seems like a communication system.
It is. If PFK is inhibited because energy is high, F6P will build up. That pushes the equilibrium of step 2 backwards, so G6T levels rise, and that buildup of G6P acts as a feedback inhibitor on hexokinase.
It's coordinated, but PFK is still the main control point, not hexokinase.
And that's because G6P is a branch point. It could be used to make glycogen. PFK catalyzes the first step that is unique to glycolysis. So by regulating PFK, you guarantee you're regulating the commitment to breaking down glucose.
And at the very end of the pathway, muscle pyruvate kinase is also inhibited by ATP, but it's activated by something from way earlier in the pathway.
It's activated by F16BP, the product of the PFK reaction.
That's feed-forward stimulation.
It is. If PFK is highly active, F16BP starts to accumulate. That signals the final enzyme, pyruvate kinase, to speed up and clear out the metabolites, preventing a bottleneck in the lower half of the pathway.
Okay, let's contrast that muscle control with the liver. The liver's job is totally different. It's focused on blood glucose homeostasis for the whole body.
Right, and biosynthesis. So its regulation is much more complex. For instance, liver PFK isn't sensitive to pH. Instead, it's strongly inhibited by citrate.
Citrate, which is an early intermediate in the citric acid cycle. What message is citrate sending?
High citrate in the cytoplasm is a signal from the mitochondria. It means the energy-producing machinery is running at full tilt, and there's an abundance of precursors for biosynthesis. The cell is basically saying, we have enough materials and energy, start breaking down more glucose.
But the master switch for PFK in the liver is this unique regulatory molecule, fructose 2.6 bisphosphate.
F26BP. This is the most powerful activator of liver PFK. When blood glucose is high, F26BP levels rise, which dramatically increases PFK's affinity for its substrate, and it completely reverses the inhibition from ATP or citrate.
So this ensures the liver rapidly soaks up excess glucose to store it.
As glycogen or fat, exactly.
The liver also has a different way of handling glucose entry, using two enzymes, hexokinase and a specialized isozyme called glucoconase.
This is critical for the liver's role as a buffer. Glucokinase has a very high KOM, which means it has a low affinity for glucose. It only starts working efficiently when blood glucose concentrations are very high.
So when blood glucose is at normal or low levels, glucokinase is mostly off, which means the high-affinity hexokinases in the brain and muscle get first dibs on the glucose.
Exactly. The liver only steps in to clear glucose from the blood when levels are soaring after a meal. It's a priority system. And glucokinase is also the primary glucose sensor in the pancreas that triggers insulin release.
What about the liver's version of the final enzyme, the L form of pyruvate kinase?
Like the muscle version, it's inhibited by ATP. But it's also uniquely inhibited by the amino acid alanine, which signals that biosynthetic precursors are available. But most importantly, the L form is controlled by hormones through phosphorylation.
So when blood glucose is low,
Glucagon is released. It triggers a cascade that phosphorylates the enzyme, which inactivates it. This is crucial. It prevents the liver from completing glycolysis and consuming the very glucose that the rest of the body desperately needs.
before any of this can happen, glucose has to get into the cell through the GLUT family of transporters. Let's quickly run through the key players.
Okay, think of them based on their affinity. GLUT1 and GLUT3 have very low km values. They're high-affinity transporters. You find them everywhere, and they provide the constant basal glucose uptake your cells need to survive.
Then there's GLUT2 in the liver and pancreas.
With its high km, it's a low-affinity sensor and makes sure those cells only take up glucose rapidly when blood levels are really high, maintaining that buffer.
And the famous GLUT4.
GLUT4 is mainly in muscle and fat cells. It has a KOM around 5 millimolar, so it's very efficient. But its action is regulated by insulin. When insulin isn't around, GLUT4 is stored in vesicles inside the cell.
But when insulin binds,
Insulin signaling triggers the rapid insertion of those vesicles into the plasma membrane, which dramatically increases glucose uptake. This is why endurance training, which increases the amount of GLUT4 in your muscle, is so good for improving glucose tolerance.
now we have to shift to a really perplexing phenomenon, the Warburg effect. Why do rapidly growing tumor cells metabolize glucose to lactate, even when there's plenty of oxygen? It seems so inefficient.
It does seem inefficient if you're only thinking about ATP yield. Complete oxidation in the mitochondria generates far more ATP, but cancer cells prioritize something else.
Building materials.
Exactly, they prioritize growth over maximum energy yield.
So what are the benefits of this rapid, incomplete metabolism?
There are two big advantages. First, all that lactate they produce is secreted, which acidifies the local environment. This helps the tumor invade surrounding tissues and can suppress the immune system. But second, and more importantly, By running glycolysis fast but stopping before the mitochondria, the cell creates a huge pool of glycolytic intermediates.
And those intermediates are the starting materials for everything a dividing cell needs.
Precisely. G6P and the others are siphoned off into pathways like the pentose phosphate pathway, which generates precursors for nucleotides for DNA and RNA synthesis, and the reducing power needed for biosynthesis. The tumor is a materials first factory, not an energy plant.
And the biochemical changes that allow this are fascinating. They express a hexokinase that resists inhibition and a slow, embryonic version of pyruvate kinase.
That M2 isozyme is key. Its slow activity creates a deliberate bottleneck at the end of glycolysis. And this bottleneck forces the upstream intermediates to accumulate, guaranteeing that they get shunted away for anabolic purposes, for growth.
This whole thing connects cancer biology with extreme athletic training through a transcription factor called HIF1.
HIF1, hypoxia-inducible factor 1. In tumors, as they outgrow their blood supply, they experience low oxygen or hypoxia. This stabilizes HIF1, which then turns on the genes for most of the glycolytic enzymes and also for factors that promote new blood vessel growth.
And the same thing happens when you do intense anaerobic exercise.
The biochemical response is analogous. Forcing your muscle into temporary oxygen scarcity activates HIF1, which boosts your glycolytic enzyme expression and vascularization. It's an ancient survival mechanism responding to a lack of O2, whether that lack is pathological, like in cancer, or physiological, like in a hard workout.
Okay, so that's breaking down glucose. Now we have to transition to building it back up. This brings us to gluconeogenesis.
This pathway is absolutely essential for survival, especially during a prolonged fast.
How much glucose are we talking about?
Your brain alone needs about 120 grams of glucose a day. Your red blood cells need another 40, but you only have about 20 grams circulating in your blood, and your liver glycogen stores are gone after about 12 to 18 hours of fasting. Without gluconeogenesis, your brain would run out of fuel very quickly.
And the primary site is the liver. The main precursors are lactate, amino acids, and glycerol.
And it's critical to remember, animals cannot convert fatty acids into glucose. Only that glycerol backbone from fats can be used.
We already said gluconeogenesis can't just be the reverse of glycolysis because of the huge negative free energy change. So it has to bypass those 3 irreversible steps.
And the first and most complex bypass is reversing that final glycolytic step, converting pyruvate back to PP. This takes 2 steps in a collaboration between the mitochondria and the cytoplasm.
It starts inside the mitochondria, step 1. Pyruvate is converted to oxaloacetate, or OAA. The enzyme is pyruvate carboxylase.
And this reaction requires ATP and uses the vitamin biotin as a carrier for activated CO2. But there's a crucial regulatory insight here. Pyruvate carboxylase is completely dead in the water unless acetyl-CoA is bound to it.
Why acetyl-CoA?
Because acetyl-CoA signals that the mitochondria have plenty of fuel. Gluconeogenesis is incredibly expensive. The presence of acetyl-CoA is a report from the mitochondria saying, "We have abundant resources. You can afford to spend the ATP to build glucose for the rest of the body."
So now you have OAA, but it's trapped inside the mitochondria. The next enzyme is in the cytoplasm.
So we need a shuttle.
Yeah.
OAA is temporarily reduced to malate, which can cross the mitochondrial membrane. Once in the cytoplasm, it's immediately reoxidized back to OAA, and this conveniently generates an NADH right where it's needed for later steps.
So once OAA is in the cytoplasm, step 2 is the conversion to PP, catalyzed by PPC.
Phosphoenolpyruvate carboxykinase. And this step uses a molecule of GTP as the energy source, and it releases the CO2 that was just added in the first step.
So this 2-step, 2-compartment strategy is how the cell gets over that huge energetic barrier.
Yeah.
It costs an ATP and a GTP just to get past that first block.
Right. And once PEP is formed, the pathway pretty much runs backward through the reversible steps of glycolysis until it hits the next block.
The PFK reversal?
Bypass 2. Instead of a kinase adding a phosphate, a phosphatase removes it. The enzyme is fructose 1,6 bisphosphatase. It just hydrolyzes F1-6BP to F6P.
And the final bypass, bypass 3, is reversing hexokinase, going from G6P back to free glucose.
This is catalyzed by a glucose-6-phosphatase, and this enzyme is the absolute gatekeeper for blood sugar. It is found only in the liver and kidneys, the 2 organs responsible for maintaining blood glucose for the whole body.
And its location is really specific.
Yes, it's bound to the membrane of the endoplasmic reticulum, the ER. G6P is shuttled into the ER lumen, the phosphatase hydrolyzes it, and then the free glucose and phosphate are shuttled back out. This compartmentalization ensures the glucose produced for the rest of the body is carefully managed.
Let's talk cost. Glycolysis nets us 2 ATP. How much does it cost to build one glucose from two pyruvates?
It is extremely expensive. It requires 6 high-energy phosphate bonds, 4 ATP and 2 GTP.
So it costs us 4 more NTPs to build glucose than we got from breaking it down. Why the premium?
That extra cost is the price of control and efficiency. The hydrolysis of those 6 NTPs is what makes the overall free energy change of gluconeogenesis highly favorable. You pay the energetic price to ensure the process only runs forward when it's absolutely needed.
Which brings us to the integration of the two. Reciprocal regulation. The cell has to prevent that futile cycle where both pathways run at once.
And that regulation is coordinated at those 3 irreversible control points. High energy signals, high ATP, high citrate inhibit glycolysis and stimulate gluconeogenesis. Low energy signals, high AMP, do the opposite.
And the major coordination switch in the liver is mediated by that unique molecule, fructose 2.6-bisphosphate.
F26-BP. It is the ultimate reciprocal regulator. High levels of F26BP strongly activate PFK for glycolysis, but they strongly inhibit fructose 1.6-bisphosphatase for gluconeogenesis.
So one molecule controls both pathways in opposite directions. What controls the level of the switch molecule itself?
Its synthesis and degradation are controlled by a single amazing protein, the bifunctional enzyme. It's one polypeptide chain with 2 distinct activities. PFK2, the kinase that makes F26BP, and FD-PASE2, the phosphatase that degrades it.
So this one enzyme is a metabolic dial. How is that dial adjusted based on what the body is doing?
Hormones adjust it through phosphorylation. Let's say you're fasting. Glucagon is secreted when blood glucose is low. Glucagon triggers a signal cascade that leads to the phosphorylation of this bifunctional enzyme.
And phosphorylation flips the switch.
It flips the switch. Phosphorylation activates the phosphatase side, FB-PASE2, and inhibits the kinase side, PFK2. F26BP levels plummet. With the activator gone, glycolysis shuts down, and gluconeogenesis is turned on to release glucose into the blood.
And conversely, when you've just eaten and blood glucose is high,
Insulin is secreted. Insulin signaling leads to the dephosphorylation of the enzyme. This activates the kinase side, PFK2, and inhibits the phosphatase side. F26BP levels spike, dramatically accelerating glycolysis for storage and synthesis. It's the body's minute-to-minute control system.
And beyond this fast control, the hormones also make longer-term changes.
Absolutely. They also control the transcription of the genes. Insulin increases the production of glycolytic enzymes, while glucagon increases the production of gluconeogenic enzymes. That takes hours or days to really shift the whole metabolic machinery.
Before this was all understood, these substrate cycles, as we now call them, were called futile cycles. Why bother running both directions at once, even at low levels?
The modern view is that they aren't futile at all. They are essential signal amplifiers. They let the system respond to tiny changes in regulatory molecules with massive changes in metabolic flow.
Can you walk us through a quick numerical example?
Sure. Imagine the forward reaction is running at a rate of 100 and the reverse is at 90. The net flux is just 10. Now a signal causes a modest 20% change. The forward rate goes up to 120, and the reverse rate goes down to 72.
So the new net flux is 120 minus 72, which is 48.
Right. A modest 20% change in the enzyme activities led to a massive 380% change in the net flux of the pathway. This amplification is absolutely necessary to explain how glycolysis can increase its rate 1,000-fold almost instantly when you start to sprint.
Finally, let's talk about how the body uses these pathways across different organs. The most famous example is the Cori cycle.
The Cori cycle is all about inter-organ cooperation between anaerobic tissues, like your active muscle and liver. When your muscle is working hard and producing lactate to regenerate NAD plus MAC, it releases that lactate into the blood.
So the muscle is shifting the burden of processing that waste product.
Exactly. It outsources the problem. The liver then takes up that lactate and uses its expensive gluconeogenesis pathway to convert it back into glucose, which it then releases back into the blood for the muscle or brain to use. It shifts the metabolic debt to the liver.
And we see a similar thing with the alanine cycle.
Right. Alanine from muscle protein breakdown travels to the liver. The liver converts it to pyruvate for gluconeogenesis and safely processes the nitrogen. It's vital during starvation for both glucose production and nitrogen balance.
This specialization even extends down to the enzymes themselves, like the different isozymes of lactate dehydrogenase.
Yes, the M4 isozyme in skeletal muscle is built for anaerobic life. It's optimized to turn pyruvate into lactate, keeping glycolysis running at high speed.
And the H4 isozyme in the heart.
The heart is a purely aerobic organ. The H4 isozyme is actually inhibited by high levels of pyruvate. This ensures that when the heart sees lactate in the blood, it converts it back to pyruvate and feeds it into the citric acid cycle for efficient aerobic energy. It basically uses lactate as a fuel, sparing precious glucose for the brain.
This has been an incredibly detailed journey. We've gone from a single 6-carbon molecule through 10 precise steps, 2 stages of investment and payoff, all the way to the complex hormonal coordination needed to choose between burning and building fuel across the whole body.
The real elegance is in that control system. The 3 irreversible steps, the 4 specialized bypasses, and that F2-6BP switch in the liver. It's a system of checks and balances designed for maximum efficiency and survival.
Which brings us to our final thought. We noted that the lower part of the glycolytic pathway, the part dealing with the 3-carbon trioses, is incredibly conserved across all life. The upper half is more flexible. This suggests the triose metabolism part is truly ancient.
And it leads to a really profound question about the original purpose of this ancient core. We think of it now as an energy-generating pathway. But what if its original fundamental function wasn't energy conversion, but the biosynthesis of triose precursors?
The building blocks for things like ribose sugars,
Yeah.
which form the backbone of RNA.
Exactly. Was this metabolic module first developed to fuel life, or was it developed to build the informational scaffolding of life itself in an RNA world?
A stunning hypothesis.
Yeah.
That the foundation of all energy flow might have first evolved not to power motion, but to actually construct the molecules of heredity.
It just forces you to appreciate the incredible depth of evolutionary time underlying every single metabolic step that keeps us going today.
Indeed. Thank you for guiding us through this essential deep dive into the biochemistry of glucose.